we have applied to the NIH Brain Initiative for a grant to support the design, manufacturing, testing and distribution of two two-photon miniaturized microscopes. These are microscopes that have essentially work very similar to large tabletop two-photon microscopes that can image at high resolution the living brain. They can image activity in cell bodies and dendrites and axons. What we've done is actually shrunk this microscope down to a size where it's a couple of centimeters, if not less. And this microscope weighs less than 4 grams, and we can essentially image with almost the same resolution as one of those large tabletop microscopes, except now these microscopes can fit on the head of a mouse and get implanted and record activity from the brain of a mouse. So in neuroscience, we're very much interested in understanding how different activity patterns of cells correlate to behaviors and how they drive behaviors. In the past, people that have used these types of techniques have been limited because these techniques have required the animal to be head fixed. For example, we are very much interested in social behavior, as these behaviors are perturbed in disorders like autism, but it's very difficult to study social behavior if the animal's head is fixed. Now, with the advent of these microscopes, the animal can freely behave, and so it greatly opens up our ability to record during a lot of these behaviors that have been studied in neuroscience now for 60, 70 years, and these behaviors have become standardized, but we didn't have the ability to take advantage of them. Now we can.
I think initially it was a bit ambitious to feel like we could take something that weighs thousands of pounds in some cases and miniaturize it to the scale of a couple grams. It's certainly an ambitious project, but we sort of got small inklings of it being possible through previous publications in the literature and also through recent developments from other areas of engineering. For example, a lot of sort of technical development in LiDAR applications or for self-driving cars and other things have been sort of brought into this project through the scanning technology. We also were very fortunate to use customized lenses. The technology for developing these lenses was pretty well fleshed out for defense applications. So for us, we were able to really benefit from a lot of technologies distributed through many areas of engineering and bring them together into a system that sort of defies logic. One of the greatest parts of developing this microscope has been just how many skill sets were involved in building it. So I was very fortunate not only to work on the optical lens design alongside a lot of input from Professor Julie Bentley at Rochester University of Optics, but also on the electronic design. So I built all of the custom circuitry, and that was in very close collaboration with Dr. Daniel Aharoni here. I also did all of the mechanical development, the mechanical design, CAD design. So I think the thing that has made this project so fulfilling and so exciting is that it's not simply just developing lenses and putting them together. It's building an entire system that works as a whole.
This grant is a very highly collaborative effort. Ali Pasha Vaziri's lab is building the temporal multiplexing version of the microscope at Rockefeller, and we will be testing it. The rat scope will be tested by multiple investigators here, including Anne Churchland's lab and Tad Blair's lab. The mouse miniature microscopes have been developed not only in our lab, but in collaboration with Daniel Aharoni, who has his own lab here and who built the one-photon microscopes, but also with Matt Strawman, who's a professor at UCSD. And he will be testing the microscopes to see, one great advantage of these scopes is that you can actually image very deep into tissue, and he will be leading that effort. And Alcino Silva will be testing the microscopes to understand ways that animals link memories together in retrosplenial cortex. So he will be using and testing the microscopes to do that, and this effort has been ongoing with all these investigators all together at the same time.